Okay, hello everyone. So for this English version of this presentation, I'm going to talk about a project that I like a lot because it's multidisciplinary and also quite interesting for the content. Because as someone part of, let's say, mechatronics, dealing with living animals is quite interesting. So my name is Gurvan Jodin and I'm from ENS Rennes. And my major is more, let's say, electronics for this project, which studies actually the fish. So for this, there is one expert, which is Prof. Dixia Fan from Westlake University. He's more specialized in AI and in hydrodynamics. And also we work with, let's say, the first co-PI of this project, James Herbert-Read, who's from Cambridge University, and he's specialized in marine ecology, and more specifically about the behavior of fish and marine animals. But to do so, we need a team because there are so much work to do that we cannot do on us all. So we have, let's say, a team of very brilliant minds working with us about this. So let's start. So this is a video of a fish. So this video in black and white is actually black and white because it's filmed in the dark. And if we go back in time, this is more what we think that fish are sensitive to. There is a wake behind the fish, and we have some cues that the fish are sensitive to these cues. So let's dig a bit further about how fish can feel this information. So there are three examples of marine animals. The first one, so this fish, you can see that small white dots all over the head, which also have all over the body. So this is some kind of, let's say, biologic sensors which are sensitive to the velocity of the water surrounding him, or also surrounding it, or also in the pressure of the environment, the dynamic pressure. If we look at this catfish, we can see this kind of appendix that he has to, let's say, to be very sensitive also to the flow, and so these whiskers also are present on this kind of seal, which make me, let's say, very sensitive to the environment and able to be very, let's say, efficient in hunting other fish. So this is something that we know, but let's dig a bit further to see how sensitive the sensors are. So for this, I will talk about hydrodynamic cues. So what we define as hydrodynamic cues is actually in the wake of a fish, you have this hydrodynamic information that survive quite a long time. And when I say quite a long time, if we compare this to, let's say, humans, it's like if someone walks in a corridor, so you disturb the air, and if someone else go after him or her, like a few minutes later, it's like if you can still feel that the air has been disturbed, and so you can feel that, it was someone, or even it was this person that I know who did this let's say a few moments ago. So this is very different from, let's say, what us humans are used to be sensitive to. So, but let's dig further. So some researchers in the year 2000, did some experiments, what we call PIV, particle image velocimetry, using lasers on fish, trying not to disturb them too much. And using these lasers and cameras, they were able to record the motion of the fluid, so to have this kind of pictures, and then dealing with this to process, and they were able to compute what we call the vorticity. So it's more, let's say, to be able to see the vortices in the wake of the fish. But they didn't stop the record after the fish passed through the area of interest. They keep recording it. And let's see what they obtained. So, to explain this colorful figure, let me take some time to say that for each column here, you take what time average, space average at each instant, and this gives you 1 point of a line. So which means one line here is 1 moment. The time just after is the row below. Okay, so each column is a position and the rows are the time. And so from 0 seconds to up to 5 minutes, so they did the record. And when we see the colors, so these colors, the red is actually high velocity, high speeds, and blue, the dark blue is actually a small velocity, which is too small for the fish to be able to sense this. And we can see that even after past 5 minutes, we can still have the, let's say, information large enough so a fish can be sensitive to it. So it's like a fish can feel that someone was there 5 minutes ago. So it's like literally, I mean, to be able to sense in the past. So okay, but then other experiments have been done in the dark. So if we look at the first one in 2006, you have the small prey which moved, and then we can see the predator, I mean, following it and be able to catch it at the end. This also has been done even a bit earlier in 2001 regarding, I mean, a three-dimension trajectory path of a fish. So in gray you have the prey, which was released like a bit more than 10 seconds before, and then in white you can see that the path that the predator follows in the dark. So when you see capture here, it means that the story didn't end well for everyone. So. Okay, so fish are sensitive to this. I mean, there are cues that they may use this to swim or to make decisions. But now let's see what are the objectives of our project to go further and validate these assumptions. So the first part is to measure those cues, measure the signature, the scale, and the stability of the hydrodynamic cues that are left behind the wake of fish. Once this is done, the next step is to quantify how the fish respond to such, I mean, historical hydrodynamic cues. The last part is to see if we can disrupt, disturb, or mimic fish hydrodynamic cues, and to see actually if that's really the cues and not, I mean, something else that haven't been considered that made them do such decision. So let's focus on the first one. So here is a drawing of, let's say, the top view of a tank, an aquarium, where you have a fish somewhere, and when we release the fish, it will take a decision to go somewhere, leaving in its path some hydrodynamic cues. Using, I mean, video cameras, we are able to record the motion of the fish, to process this and then generate a database of fish and fish motion and fish posture database. We are also able to import this into CFD software. I mean, CFD stands for Computational Fluid Dynamics, to be able to estimate the wake behind the fish. But I said estimate. So we need something to validate this. So that's why we use a pressure sensors array, which are written here, but are all over the tank. This is, I mean, small electronic devices that we developed to issue some synchronized pressure signals, and then we are able to compare this the pressure signal from the database to the actual to the pressure signals that came from the simulation. So, okay, so now the fish we use was actually the striped eel catfish. So why this? I'll detail a bit later. So you can see two beautiful pictures. The first one is from the young ones, and then you have an adult here. What's very interesting in those fish is that when we look to them swimming, so once again it's in the dark here, so the fish has quite a smooth shape, which is very, let's say, good for the, let's say, to make 2D simulations, and also they use, I mean, undulatory undulatory motion to swim, which makes them, let's say, also once again easy to detect and to compute. in the CFD softwares. Plus, also they have, very interesting, social behaviors, specifically when they are young. So once again, if you want more details about this, so you have previous work that you can see in some video somewhere in the corner of your screen if you're curious to see this. So we use, a YOLO algorithm to detect the fish. Then we saw some processing and then some work we are able to import them into CFD software that we tailor to be able to compute what's behind. Okay. So, but now what about the validation? Pressure measures. So in this picture we can see 3 pressure sensors that were working for this preliminary experiments. And if we look at the results of the pressure measures of the 3 sensors, so we can see this kind of curve. So this is the pressure as a function of time. In brown, we can see the experimental measures, and the 2 other colors are more for, let's say, digital twins. If you want more information between the different of them, once again, look to the video I just talked about earlier. So right sensor, we can see that we capture, let's say, the main change, also with quite an accurate level of pressure change. So this pressure sensor was a bit further, so yes, it didn't work that much. But if we look to this one, it's quite decent, the results. So we have the main, let's say, phenomena that have been captured. We still need to work for the secondary ones. And so that's why we are now working on some data fusion algorithm. Okay, so now this is done. What's next? So we've done, experiments, numerous experiments with various fish, various motions that they did in the tank, and also we did experiments with multiple fish, one following another, for instance. When we have this, so we go through the algorithm, the pipeline I just described, that will issue 2 databases, so one about the fish motion and posture, and the other one about the hydrodynamic cues. And so then, how can we deal with this? But so using statistics, the ethologists can now work on the first objective, which, once again, I recall it, it's to measure the signature, the scale, and stability of the hydrodynamic cues. Okay, so now let's focus toward the second objective, which is to quantify the fish response to existing hydrodynamic cues. So now I talk a bit more about machine learning and some AI algorithm, the fancy name right now. So we use the database of hydrodynamic cues that we go through this auto-encoder network that will give us a latent space which actually contains compressed information about hydrodynamic cues. We will use this to see if we can learn the fish motion from the hydrodynamic cues. So the idea is to see how efficiently we can map the hydrodynamic cues from a fish to the actual posture or motion of the fish. So if this works quite well, we can say that we could conclude that the hydrodynamic cues contain the information of the fish motion that created them. Now let's focus on what's the next part. So once again we have the experiment. So we release one fish that will make a decision, go somewhere to the shelter, and we'll leave behind him the hydrodynamic cues. Then we release the second fish. The second fish will now sense this because I remind you that we are in the dark, and the water is also come from a mix from all the scent of the other fish. So the idea is the fish can only rely on the hydrodynamic cues, I mean on his sense of the hydrodynamic cues to make a decision. So we expect the fish to follow a conspecific or to avoid a predator. But once again, maybe the fish won't do this each time, so we need to repeat sometimes, I mean multiple times, the experiments. So that's why we defined the previous pipeline to be, let's say, efficient and quick, fast to be computed, so we can process, I mean, lots of experiments. So that's the first one, but then we can change a parameter. So once again, we relieve the first fish who decides to go somewhere, but then we wait. We wait more or less time to give time for the hydrodynamic cues to dissipate, to vanish, partially vanish. Then we release the second fish. The second fish will then have less information to make a decision. And so we can see with this the impact of I mean, how, let's say, about the scalability or the dynamic of the cues, and see how sensitive the fish can be. Okay, so once again we will use AI to help us conclude. So I recall this is a previous experiment I just mentioned. So we have the hydrodynamic cues generated from one fish encoded into a latent space that we use to see if we can link the hydrodynamic cues to the motion of the fish. And now let's add the other data from the hydrodynamic cues encountered by what we call the focal fish, which means the second fish. So the second fish, we go through the cues of the first one. And so once again, we will use the latent space to see if we can, let's say, map the local fish motion decision to the hydrodynamic cues that it encountered. And then by making a comparison between both, we can conclude on how the fish can use this to make a decision. To go even deeper, we have a third let's say, objective, which is actually to see if we can disrupt, or more interestingly, to mimic artificially the cues. And so to do so, we will use once again the autoencoder that we trained before, but this time we will provide it from cues from I would say artificial devices. So the devices, machines that we put in the water to generate vortices, to generate hydrodynamic cues. Once this is done, which is the first step, we will use this train network to be able to see how we can find the optimal control of artificial device to mimic or disrupt the cues. So let's focus here. So this is a tank, and we will put some actuators. So this actuator can be maybe some robotic fish, or can be, why not, I mean, fixed devices that can vibrate or move. So this is to be defined. But this, once we know, thanks to this, the previous learning of this network, once we know how we can, I mean, what the actuation of such devices and in hydrodynamic cues, we want to reverse the problem to say, no, we want to reproduce a pressure signature of a fish that we recorded. And so we need to find how we can control the actuators to reproduce this, let's say, as much as we can. And so then we can do experiment, release another real fish and see if this time this fish follows or does not follow this artificial hydrodynamic cues. So this is the end of my presentation. So once again, I'd like to say that this presentation is funded by the Human Frontier Science Program, which is, quite a very good news for us because we are quite, let's say, free to investigate this, let's say, quite risky project and scientific questions. And so far we are in the middle of the project, but we have, very promising results regarding, fish behaviors, also the pipeline dealing with the data to issue this, and the first learning of the AI algorithm, works well. So we're quite confident that we will have, interesting things to say, and so you should follow up these videos to be updated on the most recent advances on this. Thank you for watching.